Then today's lab is going to be on target deconvolution. So for the sake of this talk, it's in the sense of we have a drug and we're trying to find the protein targets. But I also want to talk a lot more broadly about how we're doing this with transcriptomics and bioassays. And it's going to be a little review from the day. Also, thank you to all the speakers today. This was an excellent setup, making all of this review instead of an overflow of new information. So yeah, with that, dive into the introduction. As with the other labs, there will be an introduction, a short break, an hour where we actually go through the code, and then a half hour of recap. As with the other labs and a lot of this conference, the whole idea here, the ultimate motivation is making better drugs more quickly and more cheaply than we could before, bringing them to patients. The role of transcriptomics in this comes a lot down to capturing a biological state with this kind of descriptive high-dimensional assay. There's many ways it comes in. Target deconvolution is kind of one example. The other talks today were all great examples. So I think the previous labs, we did a lot of working with molecules and simulating and taking molecular structures and modeling. This is kind of moving into a new regime because you can start and predict how all the pieces interact with each other with just your computer and, of course, standing on the shoulders of everyone who figured out the Schrodinger equation and approximations of it, etc. It gets harder, of course, when you bring a protein into the mix and it's suddenly protein ligand. But as you start going into protein-protein interactions, making up organelles, cells, tissues, and organisms, it becomes absolutely unfeasible from the first principles up. And we move into a new regime where we're combining assays, measurements of what's going on in the biological system with modeling to get the best downstream results in terms of understanding biological systems, predicting how they're going to behave, etc. Kind of half tempted to race through this, but on the other hand, probably somebody missed earlier talks or would like a gentle spot where they don't have to completely focus. So I'm going to go through it, and if you want a gentle spot where you're not focusing, just take it, okay? So what we're going to be talking about today is primarily in vitro cell bioassays. And so we're talking about growing cells in lab conditions, in a test tube, a well, a petri dish, etc., where we're controlling as much as possible, letting in as few unobserved causal variables as possible. This is controlled, it's scalable. Cells in a well are about as cheap of a way as we get to look at a biological system. It's something we can readily manipulate, pipette, or get the robots to pipette for us, some drug, a CRISPR guide, etc., into the well. We know the cells we put in the well. So we have a lot of control here. And then we can run bioassays to capture a snapshot of our biological information, be it the control state or the perturbed state, so that we get information about the response to our perturbation. And this is just a brief reminder of how important this is, because this comes in as still one of the early cheap stages of drug discovery, right? It's the more information we can effectively capture, effectively utilize in the early stages for any process, the more we can fail early in the cheap stages and not send doomed candidates into the expensive stages of drug discovery. Okay, so you've seen this a lot today. For our perturbational bioassay, we have cells, we add a perturbant, causing a perturbed cell state, and then we can take some sort of measurement of that. The cells, for their part, will have different facets of interest to us. Like they might be different types, maybe they're skin cells, maybe they're neuron cells, heart cells, etc. This can also be further broken down into like naturally derived ones that you can go to a human donor to acquire, or it can be things that are cancerous immortal cell lines that have a specific ID that are, as mentioned before, the same lab and maintain clonally to be as comparable as possible. And maybe also for that have some unnatural elements to them due to their affinity of growing in a petri dish that isn't perfectly representative of cells in a human body or another organism. Besides type and line, the cells can of course be healthy or diseased, can be important, that can depend upon the owner, or we frequently say we want a simple approximation of a disease. The simplest case is, of course, a genetic disease where we just have to knock out one gene to fairly well simulate the disease state. But biologists are creative. There's lots of ways that you can simulate a disease for a cell. And then, of course, you have some pretty... The perturbant, for its part, when I'm using that, the word perturbant is here to be an umbrella term for small molecules, so drug candidates, essentially, as well as any form of genetic perturbation that we can do, CRISPR knockout, activation, knockdown, etc. And I will, of course, sometimes be grouping our mock treatment or control in as a perturbation, even if we're just pipetting our control solvent in that's not expected to actually do anything. Okay. I think, honestly, today was a great example of how this comes into drug discovery. But as a minor review, these sorts of big perturbational screens can, of course, be useful in the sense of a phenotypic screen. Let's say we have a disease state and we want to try out a whole bunch of perturbants, drug or CRISPR knockouts, and see if these could restore a healthy state, untargeted screening. It can also be even a step back from this, and Anne, of course, talked about this a fair amount, where you have some sort of global profiling or interaction mapping, where you knock out all the genes and you apply this whole drug library and you look for similarities. Today, we're going to do target deconvolution, and we're going to try and keep it simple because, after all, it's a 1-hour lab. We'll have here, but target deconvolution, we have a drug, and we're trying to figure out what the protein targets are. And this comes into drug discovery. For instance, if you had something that turned up in a phenotypic screen, but you don't know its mechanism of action, it does what you want, but maybe you need to get it regulatory approval or optimize it or do follow-up experiments or track bioassays related to it. It can be really helpful if for some reason you don't have the protein it interacts with to go find it. So that's a major application of target deconvolution. And it can also be important. Let's say you already knew what your primary protein target was, and through through any of the other methods we've talked about over the course of the summer school, you've designed a molecule that interacts with it. But again, these are now very, very complex biological systems. They have much more than just your protein target. They have a variety of conditions, temperatures, other salts, other soluble molecules, etc., and subcompartments. So under these conditions, maybe there's other things that your target drug could interact with. And looking at this from a phenotypic side can help you figure out, like, okay, early flag, are there any off-target effects that are detrimental here? Getting more specific, today's lab, we're going to be looking at the drug everolimus. If anyone knows how to say that, please tell me now. I have only ever read the name of this drug. If not, bear with me. And try and figure out in vitro targets of this drug. So maybe not in a full human, but at least in human cells in a petri dish, what other proteins there's likely targets of this drug. By the way, everolimus is a known inhibitor of mTOR, which is a key regulator for growth, metabolism, death of cells, and has a lot of aberrant signaling in various types of cancer. So, give or take how this is going to work. Again, we have our setup. We are also specifically going to take a data set that has a mix of gene knockouts as a perturbant and compounds, including our target or our compound of interest. And we have in our data set then the measurements of the effect of these knockouts. So the phenotypic effect of applying a gene knockout or applying our compound. And we're going to look for things that have a similar result. Basic idea here is you knock out the gene or you block the gene with a small molecule, we expect it to have potentially the same effect. Of course, there's lots of possibility for indirect interactions and other ways that false positives can come up here. The data in question is a high throughput perturbational assay. It's done in 384 well plates, and this is only 96 in this picture. So even has cells, compound CRISPR, and controls. It has also a variety of cell types. These perturbations were applied, and then you can do various bioassays. We heard about the phenomics earlier today, and also actually, I think a whole bunch of omics throughout the day, but this one is going to be transcriptomics. So that means in the central dogma of biology, we have the DNA, we've made this copy of it, the messenger RNA, which is transporting the information to be turned into a protein. It's basically a protein recipe. And in transcriptomics, we're just measuring the abundance of each of these mRNAs across all or some of the 20,000 genes in human genome, plus or minus some details there. I don't know how many of you don't have a biology background, but if this isn't familiar, my favorite analogy here is this is like the scheduled hours of biology's major microscopic workers here. So it doesn't tell you whether proteins actually showed up to work. It doesn't tell you how efficient they were at work. It's a proxy. It tells you how often they were supposed to come to work. Nevertheless, there's some correlation there. It's something we can measure readily in a scalable fashion, and it's close to the mechanism. So yeah, it gives us a good balance of scalability and interpretability as far as omics go. And that is probably why projects like the L1000 Links transcriptomics study exist, which is the actual data we're using today. It's a subtype of transcriptomics. I think we heard today a little bit more about sequencing types that were, let's say, untargeted measurements of anything that can be going on in a cell in terms of the mRNA levels. The L1000 is, in contrast, a targeted measurement. It said, here's my 978 landmark genes. I'm going to go find those, fish them out of solution with a probe. And this probe is attached to a color. And you take your microscope and you measure the amounts associated with each color. And this gives you your abundance measurements, basically binding to your targeted probes. And these landmark genes were selected to be as good as possible for about 1,000 genes at being able to recapitulate and predict the remaining 19,000 genes in the human genome. So they already capture a lot of the information, while being overall a lot fewer measurements, therefore cheaper, more scalable, etc. And the L1000's data is the largest publicly available transcriptomics with both compounds and genetic perturbations. So there's some of the single cell work, Perturb-seq, etc., will give you much higher number of samples, but it doesn't work with compounds. We don't have any such qualifiers on the L1000. It's very diverse, it's very useful. It's also final notes about it. It's a bulk assay, so we're not separating out single cells, but everything in the well gets measured together. And again, it's plated out on 384 well plates. Okay. During the lab today, we will be doing some embedding, and I wanted to spare a brief word for the goals of embeddings and why we chose the embedder we chose. A lot of transcriptomics data can be super noisy. This is especially the case if you have some sort of single cell data, because the single cells, like, you don't get a lot of material out of a single cell. It's a little bit like taking, I don't know, a teaspoon of flour and trying to measure it on the bathroom scale. A lot of things are just a little bit too little to measure, and a lot just falls out of the data, and there's a lot of noise. And while this may be more severe in the single cell data, it's very common to most transcriptomics data. Another way you could say it is it's a super sensitive assay that picks up on everything. Also, okay, fun fact, divergence. You can very frequently cluster transcriptomics data if you're going back to a academic context by who did the pipetting or something like this. It captures everything. Okay, so we want our embedding to denoise the data. We want it to capture biological information. We may also wish for nice things like not picking up on batch effects. There'll be a slide on that later, but practically in the embedding stage, I'm more concerned about the capture. And finally, it's often, not always convenient to reduce the dimensions of the data to not have a whole bunch of features that are correlated with each other. There's a ton of options, of course. It's an active field of research for embedding transcriptomics data. You can have some sort of supervised model. Today, we're going to take the variational autoencoder. So this is doing a reconstruction task with a sampling step in the middle, which causes the embeddings to be smoother, and it also has a regularizing effect. There's a particularly probably famous model of this, SCVI for transcriptomics, which I would encourage everyone to always use as a strong baseline, at least for the easily foreseeable future. Like, it might not be the best anymore, but it's always pretty good. Okay. The very, very recent past, last year, the year before, we've seen a lot of transformers come out that are doing a predict the mask token effect, like SC Foundation, SCGPT, UCE, Geneformer. Like large language models, they somehow have very exciting results, but not quite the same as in the natural language field. So I'm excited about these, but I think there's a little room for improvement before they can be trusted in every scenario. And importantly, for the sake of this lab, they're not very practical for getting something done in an hour. So that's a major reason we're not doing them right now. Finally, I wanted to give a shout out to graph neural networks, which are a fun idea because, as Sebastian mentioned, what inductive bias do we use for transcriptomics? Like, it's naturally a set of things. You can't throw convolution at it just because the gene identifiers alphabetically sort. But we can take what we know of known biology and how things connect, what the pathways are, what proteins interact with each other, make a graph that models that, and use that to give the network a hint about how it might want to process and understand the data. So there's some cool studies there. Nevertheless, surely for the sake of simplicity, we're just doing a small vanilla variational autoencoder that will work readily with the L1000 data. Other things we're going to be doing is some handling of the batch effect and controls. Again, we have a 384 well plate here, and any given plate, any sample from any well there is going to have some similarities with the well that's, I don't know, 2 columns and 5 rows, 2 columns over and 5 rows down. It'll be ran at the exact same time, and it'll share technical effects, like it's going to probably go into the same sequencing or otherwise measurement run. It's going to share extraction conditions when you try and get the RNA back out of it. Like, it had that same, different reagents, etc., with the exact same freshness, the exact same frequently bottle it's coming out of, and all these technical similarities will be there. Which, if this was the beginning and end of batch effect, I would say the embedding step should remove it, right? Like, we don't want that. Unfortunately for us, the batch effect is also complicated by biological effects. Like, these 2 wells on that plate, they're also going to share exactly how old those cells are. They're going to have the same growth conditions, the same time since they were taken out of the -80 freezer. They're going to be measured at the exact same time, at the exact same temperature, time of day. Cells have day it is. This doesn't sound like it always, like, we talk about these nice controlled lab conditions, right? But you can never control everything, particularly if you want the lab to run 24/7 and have these huge perturbational assays, which have a lot of these different plates going through. So because of these batch effects, it's always important for these big perturbational studies to be interpreting your perturbation responses in the context of your negative controls, the things that got a mock treatment. This might, for today, it means centering on the controls, but it could also mean something more complicated, like the counterfactual of if I had perturbed these control cells, what would it look like? There's other options here, but information controls, we might as well use it. Diving into the details. Right after break, we are going to download the L1000 data. Also, I think I forgot to say, we're taking level 2. It's smaller and reduces how much time we're going to need to take downloading. And it also gives us just landmark genes, which reduces how much time we need for everything. I'll go into later sometime in the recap for things I do differently in our real analysis. All right, we import the data, explore it, visualize it some, look at structures in the data, try and normalize it. Everyone loves a normal distribution. Then we will make an embedding with a variational autoencoder, perform some batch correction, and finally get to our actual application. Here we need to make some choices. This data has a whole bunch of different things going on in it. Like, it's not like we have one measurement for the drug and one measurement for the gene knockout and we just compare them, right? We have replicates, but we also have different doses, different cell lines. So we're going to need to make some choices here, like what we're actually going to compare. Once we do that, we can actually do our similarity search and look at the results. What was most similar between everolimus and in the panel. All right. With that, any questions before diving into the lab and break?
Questions? Okay, let's head to the break and we'll be back in 30 minutes.
So, welcome everybody to the lab. I'm going to be going through a kind of slow-paced, I talk a lot demo up here in the front. I think you all have the link on the portal site, and you are encouraged to go through it on your own. If you want a more one-on-one question, Kristina will run around and answer them. But nevertheless, don't feel bad about just raising your hand and asking to the front as well. With that, I guess I'll dive right in. Like everything else on the face of the planet, we're going to need a couple packages and to import stuff and to figure out our CUDA device. I'm going to take this pause as an opportunity to give a little context I forgot to give in the prep. I think as you go through the lab, you might notice that we very frequently take a simple, good baseline for every step. So there was a lot more focus getting, okay, all the common pieces in place than optimizing any one of these. Some of this is practical. Other decisions were made to reduce the total data size we needed to work with to download or to make it train in just a handful of epochs in less than 5 minutes, etc. So some of these are practical for the lab. I really wanted to focus on making sure you know what all the major steps are. I'll try and talk through specific examples more as I go. Maybe the first one, while this runs, is the data itself. So this is a Lynx L1000 data. For those of you who aren't particularly familiar with the data, you can probably space out, but for those of you who have touched it, you might wonder about some of the decisions that were made. First of all, I took phase 2. This is not the largest. There's now a phase 3, which is bigger and greater and better, and you should totally take if you're not trying to run a lab in an hour, and if any usage restrictions work for you, of course. Another thing we made is we chose level 2. So the data is available in various levels. You have the raw data on end 1, in the, I think, aggregated and already similarly compared on level 5. We took level 2. There's various reasons here. 1, it's smaller and easier to make work in this setting. 2, I wanted to keep this lab slightly general so that it wasn't just like the best way to work with L1000 data, but you saw the various steps that go into most transcriptomic data pipelines. Logic there. I'm not saying that you should go home and always start with level 2 in this small data set. Okay. Colab is running slower than I hoped. I wonder if I should refresh this. But it shouldn't be Colab, right? Like, I don't need a fast connection to Colab. This is on Google servers. Okay, 1 second. I also have a version of the notebook saved with results. Maybe I will pull that up. Okay. Okay, if anyone wants to let me know when the Wi-Fi is running again, we can go back to the live demo. Otherwise, it's going to turn into talk through it. I hope you guys maybe have a little more luck than I do, but I don't want to burn the whole time waiting on the Wi-Fi. So we install 2 packages. This one's just one's UMAP. Everything else we need, Torch, NumPy, Pandas, etc., is all there in the standard Colab environment. Then we go ahead and download our data. Nothing particularly special to see here, except the data we chose. We have to get once the actual data, so the abundance of our landmark genes, and second, the metadata that's associated it. What was the cell line? What was the dosage? Which experimental plate was this on, etc. Once we get the data, I want to take a brief note and stop and look at the metadata and what I find interesting about it. Important column 1 is the inst ID. This is the ID for this sample. It's going to be very necessary for simple stuff like joining the metadata with the other data. The cell ID is a cell line. The det plate is which of these 384 well plates it is on. So everything that has a shared value under det plate was on the same plate. In terms of batch effect correction, this is going to be important. Like, we'll use this column to identify which controls were associated with that data. The well can also be interesting. This tells us where on the plate it was. If you want to look, if you're going to do a very, very deep dive into this data and figure out, okay, were there edge effects on the plate, then this column would come in handy. Next, I want to focus on the PERT INAME. This is a descriptive name of what our perturbant is. It's either the gene name or the compound name. There's also an ID associated with this, but at least for the purpose of this lab, a little human legibility is convenient. There's also more data on the perturbant. The dosage, if you are very reasonably wondering why it's a -666, I think, was it Sebastian said this earlier, like figure out how they have marked their missing values. Lynx decided to mark it with -666. These are NA values. We could, of course, take care of that at import, but I think it's better to know it than for me to magically make it work in the code. It's not going to be very relevant for this lab, but. Okay, what else? We have the perturbation type column. This is going to be the column that gives us a quick, easy mapping to, was this a CRISPR knockout? Was this a compound? Was this a control? Was this a treated sample? That's what I found most important in the metadata. Any questions there? It is the way that the Lynx consortium has marked the NA values. So if they didn't actually have a measurement for that value or it didn't apply, like it's a dosage of the control, it's marked with -666 because nobody cares about the concentration of the control lids.
If you have a question, you can raise your hand and I can bring the mic to you.
If this was a live demo, I would also make the point of doing something like dot tail to show you something that's not just the control DMSO sample here. But I guess I have not the luck. All right. It turns out I did bother to stop and mention that we're working with that level 2 data. So there's a reminder of some of what I'm talking about, checking if there's anything else that's important here. Yeah, maybe notably, there's lots of public information of the original description of this data, of other people using this data. So if you're interested, plenty to check out there. The next file we import is the level 2 data. This is going to have our sample IDs that match the metadata table by essentially the RIDs which match the genes, so we can figure out which of the landmark genes these are. So it's, if it wasn't the head command, it would be 978 rows by some 300,000 columns. Again, the phase 3 L1000 data is much larger, but that's inconvenient for a quick lab. Once we have this data, one of the first things I want to do, and this applies honestly to all transcriptomics, is go ahead and check the distribution. Just with a histogram, right? In this case, I subset it to make the histogram run faster. You don't have to look at everything. But what we want to know is it normally distributed or does it have a long tail like this? I would also, of course, check simple things like integers, are these counts, or is it some sort? Of course, the L1000 data was probing and it had a continuous measurement. So we have continuous but long-tailed distribution. And a lot of our ML models work better with normal distribution. So that's one thing we're going to want to handle. In fact, it's the first thing. So the very first step here is a log transformation with a little epsilon value, so we don't do log 0. And then it's also generally convenient to work with data that's centered and has the same scale. So here we just subtract the control mean and divide by the control standard. Is that right? No, sorry. Totally awake. Okay, some of these, if you go through it, they have, one sample will have in general higher abundance than another sample. Nevertheless, these are relative measurements. So this is just scaling it so that it has the same either median or some, I guess, mean, and then centering it at 0. So getting it all on the same scale. Afterwards, plotting it again. This did not result in a normal distribution, but it's bell-shaped. It doesn't have outliers in any way like the data did going into this. It is probably close enough to work with safely with MSE loss without thinking that we are sinking our analyses. Next, I'm going to take a slightly deeper dive into the data. besides just its overall distribution. We'll do some sort of UMAP clustering to identify clusters. What are the strongest, biggest patterns in data, including nonlinear patterns. If anyone has specific questions on the code, shoot, but otherwise it's just messing with it, getting it into the shape that Seaborn likes it so that I can plot it readily. Run the UMAP and then color it by the cell line. And what do you think? Does it cluster by cell line? It's a pretty strong effect on the data. Like it's not all together, but it's definitely, generally the cell line is closer to each other than anything else. But there are some remaining differences, and we can figure out what those are quickly the moment we turn around and cluster by what went on the same plate. So now, yeah.
But like for those that don't cluster very well, like where you don't see like they look more like outliers, do you want to like get rid of those or still keep them in the data?
So question is, for things that don't cluster very well, are they outliers? Do you want to keep them in the data? And you're diving right down into the details. So first of all, I would want to figure out what they are and what actually, when I work with sequencing data a lot, I don't work with L1000 data a lot, qualifier. So preparing this lab, I went down a rabbit hole. A lot of these outliers are the positive controls. So there's something with a very strong effect that was on every plate and was large enough to get the UMAP to cluster it all together. In which case, it's kind of good that it doesn't cluster with the rest. We want to see that separating off. But yeah, otherwise, the UMAP at this stage is not about what we want. I don't want the UMAP to look like anything. I want the UMAP to be true to the data, and so I learn about the data. Once I've manipulated the data more, I might have different purposes, but at this stage, it's very explorative. So we know cell type has a huge effect, and within these cell types, like this green one here, HeLa cells, I think, you can see that the experimental batch, the exact plate, also has a huge driving effect. So there's various ways you can do any form of batch correction. Like we know this is going to, that if we want to say, okay, what's the effect of this perturbant? And the effect of the perturbant is generally being drowned out by the huge effect of cell type and the huge effect of experimental batch. We know we're gonna need to do something about that, but we have a lot of choices about what we want to do. We could, for instance, do our batch normalization now, or we could do it post-embedding. For this exact data set, I don't think it makes a huge difference. You could also take something like the level 4 L1000 data and it would be pre-batch normalized and feed that into embedding and it would be great. It's probably a more thought out method than the basics we're going to look at today. But I chose to do it after embedding, and this is more for comparability with other transcriptomics data. If this was very sparse, very, very noisy single cell data, the batch correction would work better post embedding than pre-embedding. So that's why I chose to embed first. Okay, again, we've chosen to take a variational autoencoder. I'm not saying this is the best possible way, but it's a good strong baseline. In particular, again, SCVI is almost the good strong baseline for all sequencing data, but it is specific for sequencing. Thus, we're just going to train our own variational autoencoder and also keep it nice and small so it runs in a lab. Again, shout out, even if I haven't tried to use them, I am super excited about some of the foundation models that are coming out these days, the big transformers, but they shouldn't be the first thing you try, probably. Get the safe thing first, then explore. Okay, first we need to set up our data loader. This is just reading lines out of a table, nothing special there. We take and assign our whole gene normalized table to this data set. And then we randomly split it into test and or validation and train data. Does anyone see a problem with randomly splitting it into train and validation data?
Depending on what you do, you might want to split perturbation or something. You want OD or something.
Yes, if we wanted any sort of out of distribution generalization, we should think about this step a lot more. Currently, we're doing in distribution embedding. So I don't care, but you probably should care and figure out what split is important for your downstream actual task. So we do the split, we set up the actual data loaders, of course, with training and validation, set to shuffle, get a batch size. I think everyone's pretty familiar with this stuff. For the VAE, we leaned heavily on existing implementations of VAEs. It's nothing special here. It has a very small, fully connected encoder. It predicts a mean and the log of the variation. And it has a decoder. In the middle, it has the reparameterization trick. So you take your mean and your standard deviation and you sample to actually have your Z that you're going to predict from and feed to the decoder. The forward pass just strings that all together. The loss function has your classic autoencoding loss, mean squared error, and it also has this Kullback-Leibler divergence. And the Kullback-Leibler divergence is going to push your mean towards 0 for each element and your variation towards 1. It's going to make it really hard to overfit the data, basically, and bring it together into a contiguous space. Also very important for generative models, even if this is not what we're using it for, but that we could then sample from a normal distribution if we wanted to make completely new samples. We set up our training loop with get the batch, predict, loss, backpropagation, step of the optimizer, and everything that usually goes into it, the validation loss, validation data set, no backpropagation. And then we go ahead and train it. The loss goes down pretty quickly and stabilizes. If you had more data or a bigger model, that might be different. And then this is one thing that I have taken a habit of doing for transcriptomics data that I think isn't always in the machine learning first go-to pipeline. And this is because for a lot of things, a lot of transcriptomics, you don't actually want to force it into a normal distribution. Sometimes you like to leave it in the negative log binomial count data that it is and choose a loss function that's appropriate for that data type. The stats work out better. Frequently, if you can't get it, like this isn't normal. That is to be expected. Normalization rarely works perfectly unless you do extensive things like, what is this? Q plots and quantile normalization. Unless you really force it to be a normal distribution, you're probably not going to get it perfectly normal is what it comes down to. And frequently, if we have a loss function that works with data type, it's just better to use that. So now I have to find my spot. This is one of these tricks that if you're doing something with, you're not exactly sure that this is, you've got the exact distribution right or the loss function exactly right, etc., then a great thing to do is you make your predictions and you check whether your predicted distribution matches your input distribution. So, and here actually we do see that this is, it also has a little tail that's slightly off the normal distribution. It doesn't have the little hump on the side that the actual input data did, but it's close. And of course, we actually want to check exact values, not just a distribution, plot our ground truths against our predicted data, make sure that's on some sort of line that there's a correlation there, right? That our autoencoder is actually learning something. So those are just 2 sanity checks. assuming we're happy with where we are, and there's a lot, of course, you can play with here, and for real projects, you probably should play with. There's this whole hyperparameter optimization and weights and biases and improving things that can and should be done for higher stakes things. But once you're happy with it, then we go ahead and embed the entirety of our data. I feel like there was something else I wanted to say here. Brains. Okay, with that, moving on to batch correction. So this is a thing where we had all this variance shared within plates that can have a larger effect than the individual given perturbations. And the simplest thing we can do with this is try and normalize, remove this plate-based variation from the data so that we can focus on the remaining variation in the data is the perturbance. Again, we try a pretty good go-to standard baseline method here. We are going to take our embeddings, subtract the control mean within each plate from everything else on that plate, and divide by the control standard deviation for everything else on this plate. It's a little more comparable between plates than if we didn't focus it around the controls. But the idea is simply, it will force the samples to overlap within a plate. There's a whole field about how to do this, and this is by no means like a solved problem. You have things like harmony, which use clustering within the different samples and then try and make joint clusters from different data sets that overlap with each other and put these into the same space. But then there's some assumptions, like you have the same different biology in each of those clusters. And what if between data sets, like one of those clusters just isn't there anymore? It affects the differentiation of cells and that cell type disappears. Or what if it's like our perturbational data and it's not like these huge distinct perturbations, but we have these actually very small, just like 6 replicates from one perturbation. It's very hard to cluster on this in a very, very large data set. So a lot of the existing methods aren't actually focused on the perturbational data, just because of different structures in the data. And on the other hand, working in our benefit, we have controls, and these methods don't assume you have controls. So this is why I've gone to this simple center scale to do it. Definitely something like TVN, higher compute probably works very, very well. And there's some very cool ideas coming out. Okay, in terms of code and executing this, we have to go look at that perturbation type column and our det plate, our batch effect column. We're going to group everything by batch, and within each batch, we identify the controls. This is, of course, ends up being a little bit of code because compounds and genetic perturbations have the controls marked differently, but just some overhead the first time you use the data set. And then we make a mask so that we can get the controls out. Then we're later going to run this with group by. So once we have a group, so we've gotten everything group by and group by the experimental batch. And once we have that group, we're going to fit it in here for both the metadata and the actual data. Going to use the metadata to find the controls. There we have getting the control mean, the standard deviation, and the actual centering. So very, very simple, but it's fast. And again, you can generally rely on this sort of stuff to be not that bad and catch you as you go out and try more complicated methods that you haven't messed something up, right? It's a very good first go-to. All right. Small overhead of working with the data. For instance, if I could wish nice things for myself, I would generally keep the sort order the same between the metadata and the data. This wasn't the case, so we're fixing that. Just small stuff like this in the code. And then we go through and actually run the batch correlation on each experimental plate and concatenate this all into our batch corrected embedding. Then again, we put it through a UMAP and visualize it. And now it looks like this. Also, it's not shown in the lab. You're welcome to check it someday when the internet is working and it's easier to actually run this lab interactively when you go home and do it. If you're paying attention right here, I snuck in 2 steps, right? The embedding and the batch correction, and I didn't do the UMAP in the middle. You can check me, but otherwise take my word for it that the batch correction was the major driver of these differences of everything being laid on top of each other. And this is certainly not perfect. This is why I say the batch correction is an active area of research. We still have subclusters that you can see with the cell types. And you can also see subclusters for the batch. Like ideally, you would want mixing, that you're drawing things out and the first thing you draw isn't giving you information about the second thing you draw. That would be great. But it's certainly in a much more comparable state than when we started, right? We can now take and actually say, we have a way to compare orange to blue here. One thing I would definitely be interested in if I was going to work on batch correction here is not just centering it, but also aligning the variants. I went and visualized it before, and a lot of these, as you asked, were the positive controls. And the positive controls coming from different plates aren't aligned with each other. I think the UMAP exacerbates the visual effect of this. It's not quite as bad as it looks. It's not how UMAPs work, but still, it can't be great. Like, we would love to have all our positive controls in one cluster, right? So that would be a good thing if anyone wants to go out and knock that problem out. For today, however, we are plowing on, and we're going to talk about what we actually want. The application of this data, right? Like I don't think any of you wanted me to go on about distributions of transcriptomics data. We wanted to talk about how to compare a compound to a protein, right? So we're getting to this point. We're going to take cosine similarity as a metric. We're going to take our compound, check that our compound is in the data. Obviously, any compound you haven't measured isn't going to work with phenotypic measurements. You're going to have to go back to the modeling or get some friends who have a lab. And before we actually dive in, we still have some questions left. Like, we centered and batch corrected the data, but there's still, maybe I'll go back up to the table. We have a lot of questions about how we're going to aggregate this data. We have different doses, we have different cell lines. Like, what we want to get at the end of this is compound everolimus similar to gene A, B, C, D, E, F, G, and a value for that, measurement for that. But we have, I think it's for a lot of them, it's towards 100 measurements. The minimum is 6. The controls obviously way more. And they vary, of course, replicates, dose, cell line, how the perturbation time between when the perturbation was applied and when the measurement was made. And we want to make some decisions about how to handle this. And this is like a big theoretical problem that probably there's some future cool multimodal things that handle this better. For this lab, I'm just highlighting its existence. And I'll go ahead and talk about this a little bit. So let's say we have 5 cell lines. We could just average across all of them, right? Or take the median. Like, you would definitely get a number doing this. But a brief question is like, is this really what we want to do? Or is there like a very conceptual reason that maybe we should approach it differently? And I would argue that there's definitely simple reasons that we want to approach it differently. The most obvious being that the perturbation isn't necessarily going to have the same effect in each cell type. Let's say we have a cell type, and this probably isn't the exact case because this exact lab is about mTOR, and mTOR is a very important gene. But if our target gene was not quite as central, then we'd have cell types that didn't actually express this gene, where it was hanging out in the genomic library and not in the active mRNA protein pool on standby. And if you then try and target that protein, nothing is going to happen. And it doesn't matter if you're targeting it with a genetic knockout at that point or a small compound, you're expecting no effect. And why should we be averaging this sort of stuff into our data? So there's basic questions of how do we aggregate, but also what should we aggregate that you want to think about here. For the sake of the lab, I took a very simple rule of thumb and said, if everolimus has a reproducible effect, then this is probably an interesting cell type to try and compare its effect to the effect of potential protein targets in. Like if it has a clear perturbation effect, then it's obviously hitting something in that cell and it's appropriate for comparison. Does that make sense? Surely, obviously it could be automated more also with simple hard-coded things that just didn't get done, but it could also be thought out more. You could look at things like, does it have, maybe multiple of these cell types have strong effects, but they have different effects because of something else going on in that cell. And maybe you don't want to average across it, but have some sort of information fusion. There's lots of things you can think about here, but simple methods first, right? I think I didn't scroll back to the same place. So this is essentially our dosage, plotted slightly sloppily, but from 0.4 micromolar up to 10 micromolar, and trying different cell types and looking at the cosine similarity of everolimus replicates against itself. Anyone notice anything else about this plot? Like, what's up with dose 0.1? To be honest, I didn't go down that rabbit hole, but this is also highlights an important thing to think about with the L1000 data, is it wasn't exhaustively randomized. The controls are in the same place on each plate. The cell types, of course, for very practical reasons, are always on a plate together, and I think the doses are there too. If you go just a little, not into this rabbit hole, but some other rabbit holes on the data, you definitely see that there are some whole plates that failed. And most, like, not theoretical, but practical analyses could use some extra steps in here, like filtering and failing plates, where something happened. Maybe the sequencing run failed. Maybe the plate got dropped. Maybe the pipetter that was supposed to put in 0.1 micromolar of everolimus, sorry, it pulled up air instead of the actual compound. It's hard to say, but this is almost certainly not real data. And if when we're doing this carefully, excluding this would make a lot of sense. So far, it's good. Okay, then we want to run our actual similarity search. This is essentially just pulling out our genes, choosing our cell line for the compound for everolimus, selecting it, making masks, setting up a mask to get our target dosage, making this convenient to run later. Once we have our data, we aggregate what we've selected, mean aggregation, and then run a, where is this? Cosine similarity between them. So this is select the one row of compound against everything else, because this function is going to do all in all. And this is going to give us essentially a ranked or a cosine similarity for each of the knockouts in this data set against everolimus. Then I very carelessly hacked by hand some of the cells that I found particularly red over here and chose them as where I wanted to compare them, several of them, And then it's a descending sort, so highest cosine similarity, most similar thing. And on the upside, we get mTOR. So known target is recovered. Definitely good. Definitely a good sign that the whole setup is working. And we get mTOR in a fairly robust fashion at the top or very near the top in many of the most appropriate cell types there. We also get some other recurring high hits, like the RKB, and this PIK3CA is on a lot of the lists. And these can be many things. Like it could be an off-target effect, another protein that is inhibited by the drug. It could be an indirect effect, something that's in a pathway with mTOR that just produces a similar phenotype to knocking out mTOR anyways. Or it could be an indirect effect of an off-target hit, of course. So this is a candidate list. It gives us things we should follow up on. It's not a final solution. We can talk about that either now or in the recap. But that is more or less the end of the lab proper. So surely somebody has some questions.
If you have questions, raise your hand. I'll bring the mic to you. No question? Was it too hard?
Probably more fun. Otherwise, try the internet, and if we can run it now, and everybody can maybe take the time to go through it themselves. I think that would make sense.
Hi, my question is just kind of like a clarifying question about the mean calculation, because you kind of said that maybe it didn't look right, and I was wondering if you could just explain again what it is supposed to be, or if in fact it was written correctly, the normalization part at the beginning.
1 second. Oh, you're fixing my Wi-Fi. Thank you. Okay, I had just stumbled over my words. I forgot which part of the lab notebook I was explaining and started saying the center scale bit, and not that I was normalizing, that it normalizes everything so that each sample has like the same total abundance of all the genes summed up.
Just trying to find it in here. I'm specifically just wondering, like, you divide by the means of each column and then multiply by the mean of the whole matrix. Maybe could you just explain that step?
Yes, let me get this. So this step.
Yes, that step.
Okay, so this is also in there again to make this a little generalizable for things that happen in transcriptomics. I think it helps with the L1000 data, but it's probably even more important for sequencing data, because sequencing data makes it really, really obvious why this is necessary. And this is when you're running the whole sequencing experiment, like, and the L1000, you have various steps in here. Like you extracted the RNA, that had some sort of extraction efficiency. You had your library preparation where you prepare it to be measured in whichever way you're going to measure it, adding necessary dyes, adding necessary primers, etc. And each of these steps have a efficiency of some sort, and with some exceptions involving spike ends. At the end of the day, what you measure for a given sample or cell, like the absolute numbers, don't mean much, only the relative numbers. So here we're just taking and saying, let's make it, each column have the same mean, and multiplying by the mean on the end doesn't really matter. Like the models won't care about the total scale of the data. I just thought this was convenient to think about. It also gives one numbers that are like easy to say out loud if I want to say 4 or 2, but whether this was 1 or a million or anything that is consistent doesn't really matter. The important normalizing part is dividing the individual value of each of those landmark genes by the average in that column.
Okay, that does make sense in terms of scaling, like with the last multiplication by the mean. In terms of the step after with the subtraction of the mean, I guess, is there a reason for that as well? Or is that just to bring maybe closer to zero, like to center?
Yeah, it forces the mean of the data to be at 0, which is nice for things like getting a UMAP to work without being finicky.
I was wondering why you didn't do, for example, 0-1 normalization. Um,
I don't know what 0-1 normalization is. Why should I have?
no. So
Oh, you mean so that it's
everything, yeah, everything is bound between 0 and 1. Yeah.
yeah. I could have. It's not bounded this way, which I think is reasonable for mean squared error, but it doesn't. It would have been fine. Yeah.
Okay. I was wondering if just to kind of keep the distribution of the data somehow.
It would be a good, reasonable thing to have tried and seen if this looked a little better.
Hi, I was just wondering if there were libraries that will do like the batch normalization for you.
I'm glad you asked.
Good.
Yes, there are definitely a bunch of libraries. So the first very obvious thing to point out is if you just want to work with this L1000 data, they also provide it normalized, centered, forced to actually, so they do quantile normalization and they do get like a perfect, pretty normalized distribution back out. And if you just want to work with this data, that is probably the reasonable first step. Otherwise, there are a ton of libraries for batch effect correction. It was after embeddings, right? I linked a few of them. Oh, whatever. You'll find it later in here. There's a link for something like Harmony is a big one. I think 3 of the 4 ones I linked are Python libraries or R libraries. The question is just which one is the best, but you can definitely try everything where the code runs reasonably and do this. But again, the only problem I have with these libraries is that frequently they were not made for perturbational data. Like Harmony itself, I did actually try in preparing this, and it did not successfully align things because it allows on the structure of the data being large enough to, have a critical mass of things that cluster together to make the k-means working clustering part of Harmony work. And if we only have 6 replicates of a gene that's supposed to find some 6 replicates over there, and we have a total data set of 300,000, it just didn't work with that conceptual algorithm. The others I haven't tried. I think the info core was made for perturbational data. So that might be a better out of the box thing to go ahead and give a try. And the TVN works well with perturbational data.
Hey, so I had a specific question about, well, you mentioned there are methods that look into making sure that the negative binomial distribution that it is supposed to have is kept into account. I was wondering if you can dive a bit more into the ones that you're thinking of when you mentioned that.
Yeah, you just need to choose a loss function that's appropriate for the distribution. So mean squared error, of course, you take an absolute difference of 100 is always an absolute difference of 100, regardless of whether your starting value was 0 or 10,000. But that's not true to something like a negative binomial distribution. The relative differences matter, and also you have the stochastic effect at low values. PyTorch has Poisson loss, and I think negative binomial losses. The SCVI package implements almost all of them. So that would be my first reference to send you to.
Awesome, thanks.
Any other question?
Otherwise, it's getting to be a long day. Like, I don't stand up here. We can go into the recap and
Yeah, we can go up in the recap and yeah, and
cool. Okay. So the recap is half thought out, but it's reviewing what we did, which would be interesting if we'd gotten some hands-on time to go through it and I hadn't just talked through all of it. So maybe we can go fast, and then it's basically a semi-structured Q&A, and I wanted to highlight really some of the things where I did do that differently for a real study. And of course, any questions you may have. So as basic review, download the L1000 data, import it. We saw that it had a skewed distribution. It was very close to log normal, and that there were strong effects of both cell line and the experimental batch in the data that kind of swamped out the perturbational effects. We log normalized and centered the data and scaled it. Then we embedded it with a variational autoencoder, which reduced the dimensions and denoised the data. We ran batch correction, a very, very simple, subtract the control mean, divide by the control standard deviation. We looked for a biological context, so a dosage and cell line where the everolimus had a reproducible effect. And in that cell line, calculated cosine similarity to the panel of genes that had been knocked out in the L1000 data and looked at the results. As for the results, we found mTOR pretty much at the top of the list, so that's a good sign that this is all working, and recovered several other consistent candidates. But I wanted to spare thought for how we might follow up things. Does anybody have something they want to throw out there that they'd like to see us do in follow-up? Anything you learned from days of labs? So I would probably do a kind of multi-pronged approach here. One, you can go to databases of known interactions and known pathways, and you can see, like, do I already know this is related to mTOR, and might very well be an indirect effect, that it's simply in the same pathway with mTOR, in which case, no worries, it's probably just hitting mTOR. However, if something comes up that definitely isn't clearly associated with mTOR, well, there's still different possibilities. Maybe it's an unknown interactor that's in those pathways, or maybe it's an off-target effect. You guys did things like docking in the previous lab, right? Or other computational methods to check. So at this point, we have a ligand, we have a protein. So you can follow up with computational effects to at least do a first check on whether there's likely hit there from the computational side of things. Those would probably be my first go-to, not necessarily exhaustive. Okay. I would like to say I made like a simple, minimalistic, feature complete lab today, but I actually had to cut a little on the feature complete too. So it was the major, most critical prioritized steps. If we were doing our own project, we'd probably have a little more space to prioritize a few more things. So what else would we want to put in there? Yeah, so a parameter grid for the VAE and testing different hyperparameters. Thank you. We could test different data sets for sure, particularly if this was of high interest to us. Just grab everything that's publicly available.
Kind of the open question is, do you have a general way of quantifying how good your embedding is other than downstream effects?
Yes, this is definitely something that this lab was missing, that would be very, very high on this, because even the hyperparameters that you mentioned, it's hard if you don't have one number to quantify how good your embedding is. Of course, we get metrics here, like we get the reconstruction accuracy, but the reconstruction accuracy is not our downstream effects. So I would actually say the downstream effects are critical, but what we can't do is be in this situation of kind of circulating. We want to know if it interacts with these things and seeing if we recover them at the same time. Good options here are to take public databases of interactions, mark them as known interactions, and say, okay, if my cosine similarity is my sheer predictor of interaction, so the extremes, either very high or very low, and I use this as a predictor, then go calculate something like area under the precision recall curve against known databases. Post batch correction and norm whatnot is a better metric than reconstruction anyways. I think there's also room for improvement there. You could think about linear probing to various biological annotated functions. I'd love to see creative ideas in that space, actually. Cool. So there was a couple more things that I was apparently thinking about when I was preparing this. One is we didn't utilize any of the interpretability of transcriptomics. This was one of the selling points of this data, and then I ignored it. So there's various things you can do. You can actually take the transcriptomics, and these are labeled genes. We know not for everyone, like 40% have unknown functions. We heard that today, right? But 60% is still a lot to say, like maybe this pathway is knocked out, or maybe the metabolism is revved up or suppressed, or all sorts of other things. So we can actually use the raw values of the genes to sort of describe what's going on, what a perturbation effect is like, and make hypotheses in a way that has nothing to do with ML. Therefore, it wasn't in this. We can also use it in the ML though. We could have taken something like that graph neural network, which, because we do know exactly what these genes are, we can feed them into a graph neural network and give it an inductive bias, which would also help with the processing the noisy data. Not necessarily a must, but something we could have done. And I did ramble on about this a little, but we could have definitely done some sort of perturbation efficacy and filtering. Like if it's clear from the positive controls that the whole plate failed, but nothing on that plate had an effect, maybe we want to filter it out. When we were preparing this, Kristina pointed out that also sometimes the perturbations have a mixed efficiency, particularly if this was single cell data and not L1000 data. Maybe we need to identify single cells where that CRISPR didn't cut or made a heterozygous cut instead of a homozygous cut, and consider filtering those out, handling them differently. You also have guides. So I didn't really get into this because honestly, it varies between different transcriptomic structures. But if your data is great, you want to have multiple independent guides, because not every CRISPR guide for a gene is going to knock it out or down with the same efficacy. And sometimes you'll see all the replicates for a given guide don't cluster with the others. And maybe one doesn't just want to average them in. So depending upon the major goals of your project in question, you can make a lot of gains by looking at the details of your data and whether a few simple things like filtering, or at least making models that don't just average it all together blindly, can improve things. And yes, definitely the one number evaluation to say how good the embeddings are would help. Okay. Transcriptomics data. There's a lot of things that can and do go wrong, things that happen, complexities. I don't think I didn't have anything specific I wanted to say here, but this is just an encouragement to ask questions on that topic. Questions about types of transcriptomics data.
Look at the different types of transcriptomic data.
So for perturbational, well, actually, okay, first big division first. Do we have bulk data or do we have single cell data? Do we take everything that's in a well or a test tube and measure it? So extract all the RNA together, mix it all together, and then measure it, which might have things like better signal to noise ratio, but throws out all of the heterogeneity if there's subpopulations or anything in there. On the other hand, we have single cell data where we actually take and we can, with some nice fun microfluidics magic, get specific barcodes into specific cells so that all the RNA extracted from a single cell gets marked. And when it goes on the sequencer and we sequence that barcode, we know it came from the same cell. And we can also do for genetic perturbations, CRISPR-based perturbations, we can do single cell perturbations as well, which is really nice and is a really important, really scalable method right now in transcriptomics, perturbational transcriptomics. So that's the first big thing. Then we have the choice about how we're quantifying it, which is worth mentioning. This again goes into two major splits. We have sequencing and we have some sort of probe-based methods. So one, we take everything that's RNA there, maybe we try and select the mRNA and not just get drowned out. We don't want to pay for sequencing our RNA, basically. Or we take some sort of targeted probes and we pull out just the things we want to measure. As single cell samples improve, you can get some of the cost efficacy for the sequencing. Like there's not so much of a cost split between those two as there once was. But then you have all the qualifiers of single cell and low sequencing depth, etc. Here, the L1000 was definitely on the specific probe-based side of things.
I have a multi-database question, which is there tends to not be a uniform way of normalizing all of the data between labs and everything. I think one instinct is to correct per data set, but in batch correcting in that way, you can also introduce different notches that can cause embedding problems. Is there a healthy established way of doing that yet?
No. No, I don't think there's a right answer there. You can do simple things, like if you have the time and compute and set up a nice automated airflow pipeline or whatever, or Nextflow, and it's feasible to at least take some of the technical processing, like post-sequencing processing steps and unify them. I definitely recommend that where possible. It's likely going to help. That said, it can be a ton of work. Otherwise, I think the most ambitious study I've seen on integrating these is this SysVI method, where they were really claiming not just like cross data set, but cross like proteomics versus transcriptomics, and really trying to go for the extremes of batch correction. They do use some things that I briefly mentioned, like the counterfactuals, to help with some of the differences in composition of different data sets and not just smash everything together, even if they don't have the same subpopulations. But at the end of the day, no, there's not a established good method of doing this, and it's a huge problem. I am very spoiled working for Recursion with access to fairly consistent data. So I haven't recently had to look too much into it, but. Okay.
Is there a particular type of omic that more conducive type of experiment than the bulk RNA?
What do you mean more conducive to this type of experiment?
I was just wondering, like, if I'm interested by the MOA of a particular drug or whatever, like, am I better off using single cell or bulk or even, yeah.
So again, I don't know if there's a single correct answer here. I can go through advantages and disadvantages. maybe an obvious one is if you're looking at the MOA and it only affects, let's say, anaphase, I don't know. And then maybe you average across this and there's not a lot of cells in anaphase and you don't see it. But if you look at the single cell perturbations, then suddenly you can say, okay, this subpopulation of cells does have a very strong effect. So that's an argument definitely for single cell, is when you have a strong effect, and it's going to be like downstream biology. That's going to be a very important effect, right? It just might not be obvious in tissue culture yet. But if you look at the subpopulations, you can pick it up. On the other hand, bulk drives the noise down. Definitely you want to have a system where your perturbational effects can be detected. You can't have, I mean, you can hope that you're using skin cells and learning something about the brain, but I wouldn't count on it if you can get a place where you know you have the effect.
Coming back to the talk this morning by Jason, where he was showing this ice cream, data where like if you don't take into account the season, then like the more you get ice cream, the more you lost weight. And if you take into account the season, then you realize it's the complete opposite effect. I was just wondering if like bulk is not kind of maybe doing this thing where you kind of draw wrong conclusion about like the effect of the drug. While if you are looking at the heterogeneity and the different subpopulation, you might kind of get in better idea of what's actually the effect. And the effect might be like zero on some cell type, and but yet totally opposite of what you use when you get the bulk data. So I think that's where I was going mostly.
Yeah, no, I definitely agree with you there. Like you can average over and hide or even create false associations and effects by ignoring the heterogeneity. It depends upon what your compound is. Like mTOR affects a whole bunch of things, broad effects affecting every cell type, then the bulk assay is cheaper and more effective. But if it's a niche specific or time limited effect that's only a subpopulation of cells, the single cell becomes very, very important. For that, you just have to deal with the small total sequencing reads and the large high noise.
Okay, cool, perfect. Any other question?
Okay, then a few more random notes. Unless, are we done? It was until 5, right? Yeah. You guys can walk away if you're tired of listening to me. So a few notes that I want to repeat again. Do not go home and do this just like the lab. Like take the phase 3 data. It's much, much larger. If you really looked into the lab here, there was only like 52 or 54 proteins in the set, which is hardly a genome-wide comparison. The newer, bigger L1000 data is much nicer. It's larger. Okay. And also definitely consider taking the level 4 pre-batch corrected data, at least as a sanity check. They put more effort into batch correction than the center scale did. Okay. If any of you have like actual data you're working with or different applications or whatnot, it would definitely make space to talk about things that might be different. But this is going to be a, if there's questions, thing.
Sorry, me again. So I work with some transcriptomic data, and sometimes what I get is actually like or the data that I get is actually instead of one sample, it'll be like actually pooling from 3 samples, like 3 different reps. And so I just get like the log fold change like that's been aggregated. Is do you recommend using the data like process like as it is, or should I go back and like take a step back and look more at the reps themselves?
So from my ML perspective here, I always like to take the raw reps. It's like the other is anti-data augmentation. Keeping the reps is like the simplest and most trustable thing you can do in terms of data augmentation. And also the other thing about taking log fold change is it instantly, and obviously for a lot of applications you want to do that, but I wouldn't want to do this like pre-embedding or something because it's removing so like for big models with various purposes, like I don't know what you're doing. Maybe you want to do exactly this, but generically trying to make a nice ML thing. I think it's often very important because the effect of the perturbation can depend upon your pre-existing cell state. Like different time, all sorts of different things can affect that. And if you transform it to log fold change earlier, you're just committing to ignoring any differences in the perturbation that depend upon the control state or the theoretical pre-perturbation state you can't measure. But yeah, again, it's without more information. If you're doing something simple and you just want to get it done, and the right answer is to not go down every rabbit hole and make the same considerations you would of training a foundation model. So not for your case, just my general thoughts on the matter.